Now in this session, let us move on to study nucleus. As nucleus is a very complex and elaborate structure in eukaryotic cells, we are going to divide the study of nucleus into three parts. This session consists of the first part of study. Now this session would include the ultrastructure of nucleus, nuclear pore complex and its functions. Now the nucleus or the eukaryotic nucleus is the most important and highly specialized organelle. Now eukaryotic cells do have nuclear membranes which are enclosing the nucleus. You have to remember that eukaryotic cells only have nucleus, whereas prokaryotic cells have nucleoid which is not bound by membranes. Now this nucleus contains genetic information stored in DNA. The DNA forms many genes. So all genes which are contained in the nucleus are termed or they are treated as the nuclear genome. The nucleus was described first in orchids by a Scottish botanist, Robert Brown. Usually in plants, every cell has a single nucleus, but there are certain exceptions wherein one cell has many nuclei, or there are certain plant cells which do not have any nucleus. You can see couple of dividing cells in the slide. Each cell has a prominent blue stained nucleus. If you see the transmission electron micrograph, it shows enlarged view of plant nucleus, wherein you can see a large nucleus bound by double membrane. The double membrane is called nuclear envelope. These two membranes, that is outer nuclear membrane and inner nuclear membrane, have space between them which is called as perinuclear space. This nuclear envelope separates cytoplasm from the nuclear contents. The double membrane is called nuclear envelope. These two membranes, that is outer nuclear membrane and inner nuclear membrane, have space between them which is called as perinuclear space. The nuclear envelope separates cytoplasm from the nuclear contents. The nucleus also contains chromatin and one or more nucleoli. Nucleolus is the site for ribosome synthesis. Proteins present in the outer nuclear membrane help in maintaining shape of the nucleus. These proteins are also required in cell signaling and cell division. Exact proteins present in outer nuclear membrane of plants are not known. One such possible protein is DMI1. You can see this protein present in the next figure as a green ring marking the boundary of nucleus. This protein is involved in establishing symbiotic relationships between plants and nitrogen fixing bacteria. Many inner nuclear membrane proteins are known in animal cells, but in plants only few proteins are identified yet. These proteins probably regulate mitotic processes through chromosome segregation and cytokinesis. In addition to nucleolus, nucleus contains many smaller components that are collectively called as nuclear bodies. All these structures are not bound by membranes and are highly dynamic. The presence of nuclear bodies is not very specific, but it changes with every organism, and also it can vary in response to change in environment. Let us see them one by one. You can see many nucleoli in the figure. Also, you can see speckles as green structures in the figure that are spread all over the nucleus. These speckles probably store fully mature small nuclear ribonucleoproteins, that is snRNPs, and other RNA splicing components. Next figure shows you Kajal bodies as white spots. All cells do not show visible Kajal bodies. Kajal bodies are the sites for covalent modification of small nuclear RNAs and their assembly with proteins. In plants, Kajal bodies are involved in biogenesis of microRNAs and small interfering RNAs. The next figure shows you PML bodies as green fluorescing spots. They are rich in transcription factors. We know that the nuclear envelope separates nuclear matrix from cytoplasm. Still, there is molecular exchange going on between the nucleus and the cytoplasm. This traffic occurs through nuclear pore complexes present in the nuclear envelope. There is lot of similarity in structure of nuclear pore complex in plants, fungi and animals. In first figure, you can see nuclear pore complexes as small circular structures that give a punctate appearance to the nuclear envelope. Next figure shows somewhat enlarged view of the nuclear pores. Here, each nuclear pore can be divided into spokes, central plug and ring. If you magnify them further, you can see the nuclear pore like a flower with the central plug resembling the central portion of a flower, spokes in place of petals and a ring marking the boundary of them. Outer and inner nuclear membranes are fused at nuclear pores, so they are considered to be continuous with one another. The outer membrane is also continuous with the membrane of endoplasmic reticulum. In the figure, you can see outer nuclear membrane fused with the endoplasmic reticulum. This fusion of nuclear membrane with endoplasmic reticulum enables exchange of nuclear materials with the endoplasmic reticulum. It allows the nuclear envelope to grow bigger or smaller when necessary. That means it imparts elasticity to the nuclear envelope. This figure shows you elaborate structure of nuclear pore complex. It is a complex structure made up of many proteins with very typical and specific arrangement. The nuclear pore complex spans in both nucleus and cytoplasm through both membranes of the nuclear envelope. You can see the membranes as green structures in the figure. The pore complex is about 120 nanometers in diameter. It can be seen as violet structure in the figure. Octagonal rings of spoke proteins are situated near the inner and outer membranes of the nuclear envelope. The spoke proteins connect the cytoplasmic and nucleoplasmic ring. Various nucleoporine proteins assemble to form various structures like cytoplasmic ring, nuclear basket and cytoplasmic filaments. Rod-like fibrils that extend from the cytoplasmic ring into the cytoplasm are called as cytoplasmic filaments. Fibrils extending from the nuclear ring form nuclear basket. The filaments extending from the nuclear basket link neighboring nuclear pore complexes together. The pore or the central portion of the nuclear pore complex is about 40 nanometers in diameter and is composed of unstructured meshwork of nucleoporins. Small metabolites diffuse freely through the nuclear pore complex, but the flow of large proteins and RNA is selective. Meshwork of nucleoporins within the aqueous pore governs this selective transport. Ribosomes exit the nucleus by physically moving through the meshwork. This figure shows association of nuclear pore complex with various molecules. The pore complex associates with many molecules through cytoplasmic and nuclear filaments. This association allows the pore complex to get involved in many different functions in addition to protein transport. Protein transport through nuclear pore complex requires transport factors like karyopherins, which are abbreviated as CAPS. These karyopherins mediate transport of soluble proteins as well as membrane proteins. The nuclear basket is a highly dynamic structure present on the nucleoplasmic side of the envelope. The basket is involved in many functions. First function is transcriptional regulation. It is brought about by three different types of interactions. One is between the saga complex and active genes. Second is the interaction of pore complex with TREX complexes. And third type is interaction between sumo complexes and exporting ribonucleoproteins. Second function of nuclear basket is chromatin stability that is achieved by interactions with the transcription coupled DNA repair machinery. Third function of nuclear basket is chromosome handling during mitosis. It is achieved through interactions with spindle checkpoint proteins and the spindle itself. This you would be able to see in the lower central portion and lower right hand corner of the figure. The cytoplasmic filaments link these processes to the protein synthesis machinery and cytoskeleton in the cytoplasm. These filaments interact with the GLE1 dead box protein 5 to ensure close coordination between the messenger RNP export and initiation of mRNA translation at ribosomes. Cytoplasmic filaments also interact with cytoskeletal structures to direct traffic in and out of the nucleus. The network of protein-protein interactions extending from the nuclear pore complex also includes highly conserved LEM domain and SUN domain proteins. SUN domain proteins are thought to link cytoplasmic microtubules with chromatin through direct interactions with CASH domain proteins in the nuclear envelope lumen. Now, let us move to formation of nuclear pore complex. In this figure, you can see a proposed model for de novo biogenesis of nuclear pore complex. The model shows stepwise biogenesis from steps A to step G. Look at first step A. Look at first step A. Here, many NUPs like NUP84, NUP170, 157 and peripheral NUPs form various subcomplexes. These complexes are then recruited to sites on inner and outer nuclear membrane. Step B shows presence of RAN GTPase on both sides of the nuclear envelope. RAN GTPase is required to mediate nuclear localization through pre-existing nuclear pore complexes, and also it is necessary to release assembly factors and or NUPs. In step C, you can see that the proteins at pre-assembly sites on nuclear membranes bring two membranes very close to each other. Here, POMs are considered as possible mediators of this proximity. Step D shows formation of the pore by fusion of inner and outer nuclear membranes. The reticulons stabilize newly formed curvature of the pore membrane. Step E of the figure shows stabilization of pore membrane. In step F, there is formation of a stable membrane coat by employing NUP subcomplexes. Also, POMs are linked to the central ring NUPs. Then, assembly of the peripheral NUPs forms the filamentous cytoplasmic and nuclear structures. Before concluding the session, let us sum up about the nucleus. Initially, we have seen ultrastructure of nucleus. Nucleus mainly contains nucleolus, chromatin and other nuclear bodies like speckles, Kajal bodies and also PML bodies. We have seen structures of each one of them and also their functions. Then we have seen the elaborate structure of nuclear pore complex, which is the very important and crucial part of nuclear envelope. Now these nuclear pore complexes mainly are meant for transport of proteins. So we have seen how they are arranged, how they are formed and how they function for transport of proteins. Along with these nuclear pore complexes are associated with other complexes or with other various molecules so that they perform many different functions in addition to transport of proteins. Now these associations are different, including many different complexes like Saga complex or Trex complexes or Sumo complexes or different domains like Sun domain or Cash domain. And then finally, we have also seen the biogenesis of nuclear pore complex, how the nuclear pore complex is formed. This we have seen step by step in a model for biogenesis of nuclear pore complex.